The previous video showed how information in DNA is converted into the portable form of a mature RNA during transcription. In video 12.2, I'll show you how that mRNA is used to make a bunch of proteins by translation. This video and some class practice will help us accomplish these course goals. Pause the video to take a closer look. The focus of this video is translation. The reason this process is called translation is because there's a translation in the language of RNA sequence compared to the language that's in protein sequence. The language of RNA is nucleotides, and in translation, nucleotides are translated into amino acids, the language of proteins. In the details, I don't want you to lose sight of where these processes take place. Transcription and post-transcriptional modifications happen in the nucleus, and translation happens on ribosomes that are either embedded in the rough endoplasmic reticulum or on free ribosomes in the cytoplasm. A weird thing about translation is that there are only 4 nucleotide monomers in RNA, but somehow using only those 4, we have to figure out which one of the 20 amino acids are being indicated. It took a lot of imagination by a lot of scientists, but they figured out how this works. The answer is that it takes more than one nucleotide to code for an amino acid. If one nucleotide coded for one amino acid, there'd only be 4 possibilities. That's not enough for the 20 amino acids. If 2 nucleotides coded for one amino acid, there's only 16 possibilities, and that still doesn't reach all 20 amino acids. So it turns out that 3 nucleotides code for each amino acid, and there are 64 different combinations of 3 nucleotides. A triplet of RNA nucleotides is called a codon, and you can see all 64 possibilities in this chart. But there's only 20 amino acids, so this means that there's some redundancies in the code. For example, check out leucine. There are 6 different codons that all code for leucine. But here's the thing, this codon code is pretty much universal for all living things. For example, CCG is proline for every organism. Scientists take that to mean that all living things are related, since they use the same code. Also, even though the code is redundant, the code is unambiguous. This means that there's no confusion. UUC is always phenylalanine. If you look at all the possibilities, you'll see that 61 of the 64 codons code for amino acids, and 3 of the codons are stop codons. Also, you'll notice that one of the codons, AUG, is a start codon and also codes for the amino acid methionine. Now that we've seen how the code works, I'm going to switch gears here and talk about the molecular machinery of translation. Really, translation is the story of 3 different types of RNA working together to make proteins. There's mRNA, or messenger RNA, tRNA, or transfer RNA, and rRNA, or ribosomal RNA. mRNA is the RNA that we've been talking about so far in our transcription video. mRNA is the RNA that's made by transcription and those post-transcriptional modifications. The mRNA is the portable version of the gene that takes information from the nucleus to the ribosome. Now that we know about codons, we can add that the coding region of the mRNA starts with the start codon AUG and ends with a stop codon, which might be AUG or one of the other stop codons. Transfer RNA has the job of carrying amino acids. tRNA has a pretty cool clover leaf shape here, and we want to focus on the 2 opposite ends. One end is the anticodon arm, and the anticodon arm has nucleotides that are complementary to the codon on the mRNA. By matching up with the codon on the mRNA, the tRNA brings an amino acid into the mix. The amino acid is attached to the other end of the tRNA at the amino acid attachment site arm. The process of adding amino acids to tRNA, referred to as charging the tRNA, is shown in this diagram. The process requires an enzyme called aminoacyl tRNA synthetase, or more friendly version, charging enzyme. The substrates are the amino acid and the tRNA, and since this is an anabolic reaction, ATP is required to join the substrates together to make a charged tRNA product. Here's another diagram showing the life of a tRNA. In the first two panels, the tRNA is getting charged with an amino acid by the charging enzyme. Here's that charged tRNA. Then, the anti-codon arm of the charged tRNA matches up with the appropriate complementary codon on mRNA, bringing that amino acid to add to a growing protein. Our final RNA type is rRNA, or ribosomal RNA. rRNA is a huge part of ribosomes. Ribosomes are made of a large subunit and a small subunit. Both ribosome subunits are made of a mixture of proteins and rRNA. Together, the subunits act like a huge enzyme that builds proteins using anabolic dehydration synthesis reactions. Once the large and small ribosomal subunits are assembled, they form a ribosome with 3 chambers. These chambers are called the A site, the P site, and the E site. I want you to think of the A site as the amino acid site, because that's where the amino acids first come into the ribosome. Think of the P site as the polypeptide site, because that's where the amino acids are joined together to make a polypeptide. And think of the E site as the exit site, because uncharged tRNAs end up there after they dump their amino acid onto the peptide, and then they have to exit. So E is for exit. So let's put the story of those 3 RNA types together to tell the story of translation. Just like transcription, translation can be broken down into 3 phases: initiation, elongation, and termination. In initiation, it's a matter of bringing together all of the RNA types. The ribosome will bind to the poly A tail of the mRNA and then slide along the mRNA sequence until the ribosome reaches the start codon. At this point, the tRNA with the complementary anticodon, UAC, is brought to the P site of the ribosome. This complete complex of ribosome subunits, mRNA, and starting tRNA are called the translation initiation complex. Once everything is assembled, it's time to make the amino acid chain, the polypeptide. At this point, the tRNA with the matching anticodon enters the A site. This is the next tRNA, bringing the next amino acid in the chain. Once those amino acids are close, now the ribosome can transfer the amino acid from the tRNA on the P site to the tRNA in the A site that's shown here. Next, the ribosome slides over, shifting the sites of the tRNA. The tRNA that was in the P site shifts to the E site, and the tRNA that was in the A site shifts to the P site. The A site is open again. Now, the uncharged tRNA in the E site will exit, and then the next tRNA with the appropriate anticodon will come into the A site. Once that tRNA comes into the A site, the growing peptide chain will be transferred over onto that tRNA, and this process will cycle through again and again and again, growing the polypeptide chain 1 amino acid at a time. Termination happens when a stop codon enters the A site of the ribosome. There's no tRNA with an anticodon to a stop codon. Instead, a protein called release factor now enters the A site. release factor triggers the release of all of these components from one another: the polypeptide, the uncharged tRNA, the ribosome, and the mRNA. The mRNA can be translated again to make another protein, maybe by the same ribosome, but probably by another ribosome. tRNAs need to be recharged with another amino acid, and the peptide needs to be processed. I've intentionally been using the word peptide and now not protein, and now I want to clarify. A peptide is just a linked together chain of amino acids, but peptides are not proteins yet. To become a protein, a peptide must be folded and modified. Folding and post-translational modifications give a protein the right shape and chemical properties to do its intended job. Peptide folding actually starts before the peptide is fully made. You can see that happening in this picture. This shows a ribosome that attaches to the rough ER. The white dots show the growing polypeptide chain. Once the chain starts coming out, the chain starts folding, adding secondary and tertiary protein structure. Additional modification can happen once the peptide is inside the endoplasmic reticulum. Another cool thing about proteins is that the first 20 or so amino acids that are indicated in red here are called the signal peptide. The signal peptide acts as a signal for where is the protein supposed to end up in the cell. There's another protein called signal recognition particle, or SRP, that recognizes and binds to the signal peptide and acts as a chaperone to chaperone the peptide while the peptide is still being made to the destination where the protein needs to end up, in this case, the rough endoplasmic reticulum. The last two videos have covered transcription and translation. I didn't need to make separate videos about how humans do these processes compared to yeast or compared to fruit flies, and the reason is because all living things use the same processes. It seems unbelievable that every living thing on Earth, as diverse as organisms are, use the same 4 nucleotides for genetic material, the same 64 codon code to code the same 20 amino acids, and even the same machinery to build and move this information around. The similarity of the building blocks and processes are really strong evidence that all organisms on Earth originate from a common ancestor. Let's recap what we've learned in this video. Translation is the main focus of this video, which converts information in RNA to a peptide sequence using ribosomes. We learn about how the triplet codons are needed in mRNA to code for each amino acid. And there's 3 different RNAs involved in the translation process. mRNA provides the code, tRNA transfers amino acids to a growing protein, and rRNA acts as a part of the ribosomes. We divided translation into 3 phases to be able to understand what's going on in those phases, and then talked about how translation makes kind of pre-proteins that still need to be folded and modified to become fully functional proteins. And last of all, we connected this to evolution by talking about the fact that the universality of the nucleotide building blocks and the amino acid building blocks, as well as the universality of the machinery that's used to make all of these things, is really strong evidence to say that everything on Earth has derived from a universal common ancestor.